With single cell analysis, every single cell matters. And that's what makes it difficult, because if you do bulk experiments and some cells have low quality or are lost during the process, it's okay. You still have the whole bunch of other cells. But here, every single cell matters, and that was always a hurdle to get every cell being of a representative quality and being representative for the biological question you were asking. I'm Sarah, working in the TechWatch team as a technology specialist. That means that we scout and de-risk technologies, novel technologies that are just brand new on the market. In life sciences, if there are new technologies coming out that could be breakthrough and it could help the scientists within the VIB, so the Flemish Institute for Biotechnology, then we reach out to the companies and then we eventually try to get the technologies in-house for testing and evaluating if the technology could be a breakthrough technology for our institute.
The Lightcast technology was already on our radar for quite a while through patent search and literature search. So we noticed already a few years ago that there was quite a lot moving in the single-cell functional analysis domain, and Lightcast was one of the names that popped up. A few months ago, we finally got the prototype platform, which we're very happy for. And now we're actually in the process of running samples and trying to fit some of the research samples onto the platform.
TechWatch are, quite frankly, the best in the world at doing this sort of collaboration. They have a huge amount of experience working with beta and early access instruments. They pretty much get through almost all of the new instruments that come out. They've worked with them. They have seen them from a very early stage, and they have helped with that development. My job is to introduce them to the instrument, train them on it, but also come back frequently and help them to actually design their experiments, figure out exactly how to do what they want to do with the instrument. We can talk to them, we can see what are the problems they're really trying to solve, and does this solution do it in the best possible way, or is there something we should be changing? Sometimes we will actually do this sort of thing and learn about a problem we didn't even know existed that it turns out our technology is the best solution for.
All of the single-cell genome and transcriptome and epigenome is a snapshot. Of course, you can do time point experiments, but it's not that you see your cell behaving and being dynamic. And with these kind of platforms, that are the kind of questions then you can add on top of those layers. You can first look at your cell, how it behaves, how it interacts, how it reacts to components, and then you can get the cell out of the platform and still do all of these snapshot technologies.
There are a number of products out there that can look at single cells. Many of them are droplet-based, some of them are well-based, some are pen-based. So these already exist, certainly. The trick with all of those is that they are very limited in terms of being able to get single cells into those droplets or wells or pens in a reliable way. And that is where we make a big difference. It's unprecedented control over the actual droplets themselves. By being able to move them with light, move them into an array, put them wherever we want to put them, and make them interact with whatever other droplets we might want them to interact with. This is something that no other system can do, and it allows us to be certain that every single droplet we are looking at actually has the cell of interest in it.
Technologies such as Lightcast, they enable to basically keep your cells alive, track their behavior over time, do treatments, find out what the heterogeneity of the treatment response is, even in a time course fashion, and then pick out cells of interest to further do studies downstream. That aspect is really filling a gap now in the research needs. So that's where we try to plug in. There are different projects both in the cancer area as well as in the neurobiology area where these type of technologies would fit in. And so the fact that they have developed this optical EWOT technology as such, it's really one of a kind.